The word biomacromolecules signify the large molecule which is present in a biological system. As we know that big wall of house is made up from bricks. Similarly, biomacromolecules are large biological molecules assembled from relatively simple precursor, generally called as monomer. Each monomer are linked together by a respective strong covalent bond and form a long polymer. Therefore, biomacromolecules are also known as biological polymer. The number of monomers in a biomacromolecule can range from tens to millions, and therefore they have high molecular weight. Biomacromolecules may assemble together into a complex supramolecular complex. This supramolecular complex play a crucial role in biological processes. For example, ribosome, a protein synthesis machinery, is made up of 70 different proteins and different RNA molecules. Biomacromolecules are major constituent of cell. For example, in E. coli, 70% of water and 30% as a solid material. Out of that, protein constitute 50% and nucleic acid constitute 23.3% and carbohydrate constitute 10% of the cell's dry weight. The term macromolecule was coined by the Nobel laureate Hermann Staudinger in the year 1920s. Now the next question is, what are the major biomolecules? Generally, there are four major classes of biomolecules, namely protein, nucleic acid, polysaccharide and lipids. Now let us learn each of these major biomolecules one by one. Number 1, proteins. Proteins are long polymers of the monomeric unit called amino acids. Therefore, amino acids are known as building blocks of protein. In protein, each of the amino acids join together and form a long chain by a special type of covalent bond known as peptide bond. It is also known as amide bond. In reality, the protein molecules often contain more than 100 amino acid units. For example, human cytochrome C is a small protein that has 104 amino acid residue. Its protein differ widely in amino acid composition and also their amino acid sequences. Proteins are present in all compartments of the cell and constitute 50% of cell dry weight and hence occupy the largest fraction among the biomacromolecules. Proteins are most versatile. They may act as enzymes and also serve as structural element of a cell, signal receptor, transporters that carry specific substance into or out of the cell. Now let us discuss the general structure and properties of amino acid, the monomer of protein. From the picture, we know that all the amino acids have two important functional groups, namely carboxyl group or COOIs and amino group, the NH2 group, and a side chain R group, which is differ in their structure for different amino acids. The amino group and the carboxyl group are highly reactive and involved in the peptide bond formation. However, R group is not involved. Even the presence of the functional group such as COOIs and NH2 group. There are about 100 amino acids have been shown to present in various plants and animals. However, only 20 of which have the L isomer are found as a constituent of the most proteins. These 20 amino acids are differ only in the structure of their side chain, the R group, and they are often referred to as standard amino acid. And two amino acids, namely cellulose and pyrrolysine, was newly reported to found as constituent of protein. Cellulose is present in several enzymes such as glutathione peroxidases, thioredoxin reductases, glycine reductases, whereas pyrolysin in some methanogen bacteria. Now, let us learn how the protein is made from their amino acid monomer. Formation of peptide bond is an example of condensation reaction involving losses of water molecule. In this reaction, alpha carboxyl group of one amino acid, say R1, is reacted with the alpha amino group of another amino acid, say R2, and the two amino acids are joined together by a covalent bond known as peptide bond. The resulting product is called dipeptide, which contains two amino acid units. The peptide made up from 2 to 10 amino acids are called as oligopeptide. The proteins are polypeptide with more than 100 amino acids. The word protein and polypeptide is used as an interchangeable term. The bond between two adjacent amino acids in a protein is a special type of amide bond, CONIs, where the carbon atom of COOH group is linked with nitrogen atom of NH2 group of the adjacent amino acid. The repeated amide nitrogen and alpha carboxyl and carbonyl carbon atoms of each amino acid residue form the backbone of the protein chain. Proteins differ in amino acid composition and their sequences. They may also contain one or more polypeptide and can be linked by disulfide bond in addition to peptide bond. For example, serum albumin, a human protein, is a single polypeptide chain and has 609 amino acid residue, molecular weight of 68,000. Human insulin has two peptide chains containing 51 amino acid residue linked by a disulfide bond. Glutamine synthase in E. coli contains 12 polypeptide chain that has 5,628 amino acid with a molecular weight of 6,19,000, whereas chitin, a human protein, is a single polypeptide chain that has 26,926 amino acid with a molecular weight of 29,93,000. Now, structural and biological properties of peptide bond. Peptide or amide bond has a partial double bond character and is always in a trans configuration. They are rigid and lies in the same plane where the side chain are protrude out from the primary chain. Peptide bonds present in a proteins are stable in physiological pIs. Now, N and C-terminal amino acid. Each amino acid in a protein or polypeptide chain is termed as residue. The terminal amino acid residue with a free amino group is called N-terminal amino acid, and the one with other free carboxyl group at the other end called C-terminal amino acid. By convention, the N-terminal is taken to be the beginning of polypeptide chain. Thus, in the tetrapeptide tyrosine, glycine, lysine and alanine, tyrosine is the amino terminal residue and alanine is the carboxyl terminal residue. Now, covalent bonds involved in the protein structure. The principal linkage found in the all protein is the peptide bond or amide bond. In addition to peptide bond, a second type of covalent bond known as disulfide bond is found in the proteins and polypeptides. Disulfide bond join two cysteine residue that present in a primary chain to yield a 1 mole of cysteine. Cysteine is an amino acid with a disulfide bridge. Now, the second major important biomolecule, nucleic acid. Nucleic acids are polymer of monomeric unit known as nucleotides. There are two types of nucleic acids, namely deoxyribonucleic acid or DNA and ribonucleic acid RNA. Let's discuss on structure of the nucleotide. Each nucleotide is made up from 3 small subunit molecules. Number 1, a nitrogenous base. It may be either purine or pyrimidine. Number 2, a pentose sugar, either deoxyribose that present in DNA or ribose that present in RNA. And number 3 is a phosphate molecule. The adenine and guanine are derivatives of purine, whereas cytosine, thymine and uracil are derivatives of pyrimidine. A molecule containing sugar and nitrogenous base without a phosphate group is called as nucleoside. Structurally, in each nucleotide, the carbon 1 position of sugar is covalently linked at N1 position of pyrimidine and N9 position of purine by a glycosyl bond, and phosphate is covalently linked to the 5' position of pentose sugar as a phosphoester bond. The formation of N-glycosyl bond and phosphoester bond is favored by removal of water molecule, a hydroxyl group from the pentose and hydrogen atom from the base, and phosphate. Now the types of nucleotides and their representation. There are 4 types of structural units known as deoxyribonucleotides or deoxyribonucleoside 5' monophosphate. They are written as DAMP, DGMP, DTMP and DCMP in DNA, and 4 ribonucleotides or ribonucleoside 5' monophosphate written as AMP, GMP, UMP and CMP in RNAs. Now, formation of nucleotide chain. The successive nucleotide of both DNA and RNA are covalently linked to a phosphate bridge, in which the hydroxyl group of 5' phosphate of one nucleotide unit is joined to 3' hydroxyl group of the next nucleotide, form a phosphodiester linkage with the elimination of water molecule. The polymerization of primary structure of nucleic acids proceed in 5' to 3' direction by joining of individual nucleotides into the existing nucleotide. Thus, the covalent backbone of nucleic acid consists of alternating phosphate and pentose residue, and the nitrogenous base may be regarded as side groups joined to the backbone at the regular intervals. By convention, the primary structure of DNA or RNA molecule is written as 5' to 3' direction. DNA or nucleic acid containing 50 or less is generally called as oligonucleotides, and the longer nucleic acid is called as polynucleotide. Like polypeptide chain, primary structure of polynucleotide chain has 2 polar ends called 5' end and 3' end. The free 5' hydroxyl group attached to a phosphate is known as 5' end, whereas the other end that has free 3' hydroxyl group attached to a pentose sugar is called as 3' end. By convention, the base sequence is written in the 5' to 3' direction. The backbone of DNA and RNA are hydrophilic, and hydroxyl groups of the sugar residue from the hydrogen bond with water. The covalent backbone of DNA and RNA are stable and therefore unsusceptible to non-enzymatic hydrolysis. Now let us discuss the third type of biomolecule, polysaccharides. Polysaccharides are polymer containing more than 20 monosaccharide units, and some have hundreds or thousands of such units. They play a vital role in energy storage and in maintaining the structural integrity of an organism. Polysaccharides are also simply known as glycans. The monosaccharides are either aldehyde or ketones with 2 or more hydroxyl groups, Some common examples of monosaccharide are glucose, fructose, galactose, ribose, etc. Polysaccharides are formed by joining monosaccharide units through O-glycosidic bond or linkage. In maltose, 2-glucose residue is joined by glycosidic linkage between C1 of anomeric carbon atom and C4 of another glucose molecule. This type of linkage is known as alpha 1 to 4 linkage. Polysaccharides are two classes based on their type of monomeric unit that make up. Number 1, homopolysaccharides or homoglycan, and number 2, heteropolysaccharides or heteroglycan. Now let us discuss on homopolysaccharides. It contains single type of monosaccharide molecule. For example, starch is made up of only monomeric unit of alpha D glucose. This alpha D glucose residue are joined by alpha 1 to 4 linkage in the amylose, the unbranched form of the starch. However, it also contains alpha 1 to 6 linkage at about 24 to 30 residue in addition to normal alpha 1 to 4 linkage in amylopectin. Similarly, glycogen is also a homopolysaccharide made up of repeating unit of alpha D glucose. However, it has higher degree of branching than the starch. Most of the glucose units in glycogen are linked by alpha 1 to 4 as a glycosidic bond, and the branches are formed by 1 to 6 glycosidic bonds present about once in 8 to 12 units. Now cellulose. Cellulose is a linear homopolymer of beta D glucose unit linked by beta 1 to 4 glycosidic bonds and present as a major component of the cell wall in plants. It is one of the most abundant organic compounds in the biosphere. Next is chitin. Chitin is a linear homopolysaccharide composed of an acetyl glucosamine residue in a beta 1 to 4 linkage. It is the principal component of hard exoskeleton of nearly 1 million species of arthropods, for example insects, lobsters and crabs. And the second type of polysaccharide, the heteropolysaccharides. Heteropolysaccharides contains 2 or more different monosaccharide units. Some important examples are peptidoglycan. Peptidoglycan is an alpha 1 to 4 link polymer of N-acetyl glucosamine and N-acetyl muramic acid units joined to a tetrapeptide. Glycosaminoglycan is a linear chain of repeating disaccharide unit in which one of the monosaccharide unit is an amino sugar and one or both of the monosaccharide units contain at least one negatively charged sulfate or carboxylate group. For example, heparin, hyaluronate, chondroitin, keratin sulfate, etc. Number 4, lipid. Lipids are chemically diverse group of compounds. They are hydrophobic in nature and have various biological functions. For example, fats and oil serve as a storage form of energy in many organisms. Phospholipids and sterol are major structural elements of biological membrane. Other lipids play a crucial role as enzyme cofactor, electron carriers, hormones, light absorbing pigment, etc. Individual lipid molecules have molecular weight in the range of 750 to 1500 Dalton, which is smaller as compared to other macromolecules such as protein, nucleic acid and polysaccharide. Therefore, they are not classified as biomacromolecules. However, many lipid molecules may aggregate non-covalently and form a large structure. For example, cellular membranes are built up from millions of lipid molecules non-covalently. Sometimes they are not considered as true biomacromolecules due to lack of the covalent bonding. Conclusion: biomacromolecules are large molecules found in biological system. They are assembled from simple precursor of biological molecule having molecular weight of 500 or less and generally called as monomers. Protein, nucleic acids and polysaccharide are important major biomacromolecules which are made up of amino acid, nucleotide and monosaccharide units respectively. The number of monomer units can range from 10 to millions, therefore, they possess high molecular weight. Each monomer unit are joined together and form long chain of polymer by a variety of respective covalent bonds. For example, amino acids are joined by peptide bond in protein chain, nucleotides are joined by phosphodiester bond in nucleic acid,